sure that's recording. It is. Good. Right, so we go to share screen. Okay, so this is going to be a little review on hydrodynamic methods. These are methods for looking at molecules in solution. So it completes the solution loop, if you like, in terms of what we've been talking about with the properties of molecules, including molecular weights and conformations you've just been talking about. But 4 techniques: the analytical or centrifuge, SECMALS, dynamic light scattering, that's what DLS stands for, and viscometry. We've done that in detail already, but I'll put it here because it links nicely in with the light scattering. SECMAL stands for size exclusion chromatography coupled to multi-angle laser light scattering. SECMALS. Now, what's happening here? Right, so yeah, that's the NCMH. That's where we're going to at the end of this, just to see some of the techniques being used. We've got an experiment running at the moment on glycopeptide psychoplanin, so we can see what's happening there. So we'll go along after we finish this brief introduction. So these are some of the hydrodynamic methods. These are the principal ones, if you like, that we have here. There's the analytical ultracentrifuge. It's a high-speed ultracentrifuge. High speeds meaning speeds up to 50,000 RPM, which is about 200,000 G, with an optical system, so we can look at the sedimentation properties of molecules and make deductions from that. There's classical light scattering, multi-angle light scattering, where we look at the scattered intensity envelope as a function of angle, formed by macromolecule scattering in solution at different concentrations. And by looking at the scattering patterns at different concentrations, different angles, we can get information about the molecular weight and conformation of a molecule, similar to what we can do with the ultracentrifuge. Then there's dynamic light scattering, where we look at the fluctuations in intensity caused by of light scattered caused by the motions of the molecules, and the motions of the molecules will be related to their size and conformation. This is another method of getting conformations, particularly of larger macromolecular assemblies. And finally, viscometry. We've already covered that in some detail, but I'll show you how this is coupled to the SECMALS. So it's a way of looking at the viscosity of solutions which are mixtures, which have got other molecules that are present. Maybe contaminants, or maybe it's a polydispersed solution. But we can couple that with the size exclusion chromatography, which is already linked up to the light scattering. Right, just briefly, dynamic light scattering first, and we put our solution in a cuvette. Some DLS instruments, you inject the solution through an injection port, but in the Malvern nanosizer or zetasizer system, which we've got over in the NCMH, it's cuvette based. And these cuvettes and solutions have to be scrupulously clean because of the problem of stray scattering from any aggregates or dust particles or things that are present. That being said, the software now is very good at filtering away the contributions from large supramolecular aggregates, which can obscure the light scattering signal from your molecules. But it's important that sample clarity is maintained, and this is the idea. You got a light beam impinges on your solution of molecules, which are moving around due to Brownian motion, thermal motion, at a rate depending on the size and the shape. Generally, the smaller molecules tend to be more nippy. They move around faster, and also the compact molecules move faster because they experience less frictional resistance as they're moving through the solution. And how fast they move impacts on the fluctuation rates of intensity. By the way, has Vincenzo already given you some stuff in Gleb's module on dynamic light scattering or not? Not done that yet. Okay, so this is a nice introduction to that. He'll go into probably more detail. So, unlike a spectrophotometer where you're looking at the loss of intensity of the incident beam caused by absorption, UV or visible absorption of molecules, I mean, you can use this also to look at light scattering as well from the so-called turbidity of the molecular solution, because you will get loss of the intensity light through scattering, and that sometimes has to be corrected when you're making absorption measurements on solutions. It's not usually a problem at dilute solution, but it is at higher concentrations. Then at high concentrations, you can't use absorbance because of the limits of the Lambert-Beer law. Now we're interested in looking at the intensity at angles away from the incident as measured by the scattering angle theta, and the fluctuations intensity caused by the motions of the molecules, which are linked to the what's called the Doppler effect. And a detector system coupled to a correlator computer. Correlator correlates the changes in intensity with time. Too rapid for the eye to see. There's the microsecond, nanosecond types of fluctuations, but the correlator can pick this up, and by comparing the change in intensity with a reference time, we get what's called autocorrelation plots, and from these autocorrelation plots, we get a measure of how fast the molecules are diffusing, as measured by what we call the translational diffusion coefficient, which is related to particle size and conformation. This is what a setup used to look like. This was after I started here in the 1980s. This was one of the first instruments that I acquired, helped by generous donations from various companies like Unilever. We got this set up. So here's your laser here. This is where your solution goes in here, and this is it's in a water bath at constant temperature. And this is the detector system at an angle of 90 degrees. This is the goniometer system, so you can change the angle. So laser, helium neon laser, the solution here, and the detector, and then this signal goes to this big box here, which is an autocorrelator, and then the data is analyzed by this computer setup. This is what a DLS, or its real name, quasi-elastic light scattering setup, used to look like. And you can see from the age of the computer here, this IPC computer, you can see this is pretty old. This picture was taken in the late 1980s. But this instrumentation's been superseded by this. Everything's got smaller, lasers become laser diodes and all that sort of stuff, and it's great in a way, because from a health and safety point of view, everything is boxed in. And when we did this, it's all sorts of safety controls because of the laser. The experiments were done in the dark because we frightened of stray light interfering with the measurements. If someone put a switch on or something in the next lab, that would send a pulse through and would disturb the measurements. So that was a different time, different sort of ethos towards doing experiments. And then this type of instrumentation appeared after the 1990s. This is the Malvern nanosizer, Zeta sizer. It's quite small, so everything is boxed in here. The laser and the detector, and this is where the sample goes in this box here. And that's the switch. This is all linked up to a computer. It's all computer controlled. That we'll see when we go over to the NCMH in a minute. And these are the sort of plots that come out. You get, well, it's a diffusion coefficient, what you measure, but the software automatically converts this to a size distribution. And you can see you've got some macromolecular stuff here, and also some very high molecular weight material here. So it's a good way of assessing quickly. It's a very rapid technique, DLS. You get results within minutes. The quality of your solutions, and because it's so rapid, people use this type of technique for crystallization trials to see if various formulations will promote crystallization, nucleation of important types of molecules which need crystallizing. So it's a useful technique to have for a wide range of applications in biopharma, because of its rapidity, and even in crystallization laboratories. And then a variant of this is the electrophoretic light scattering, where you have these special cells where you can basically place a charge across the solution, and this is what we call electrophoretic DLS, and it's used for measuring what's called zeta potentials of solutions, which is related to the charge on the molecule. It also depends on mobility parameters as well, but it's related to the charge on a molecule. So that very briefly is DLS. SecMals, I mean, we'll be using this when we do the group mini project, so you'll see how this works in some detail, and the precautions we take to make sure we have clear solutions. But SecMals, this device here, this blue box, actually this is an older version of our instrumentation, but that's what's called a light scattering photometer, and it measures light scattering at multiple angles. That's why it's called MALS, multiple angle light scattering. We used to call it multi-angle laser light scattering, but now since virtually all light scattering instruments have a laser, the lasers become superfluous. So we just call it MALS because it goes by understanding that there is a laser involved. This is the MALS device, and it's coupled to SEC, size exclusion chromatography, which is enclosed in this temperature cabinet here. Now, if we take the lid off one of these light scattering photometers, we'll see this, and basically you have, and as we'll see when we go across later for the demonstration, what happens is that solution comes from a chromatography column and is fed into this flow cell. And as it flows through very slowly, a beam of laser is shone through the solution, and whilst the solution is moving through slowly, and when it comes up a column, you have the larger molecules coming off first and the smaller molecules if you've got a mixture. As it comes through, we get these basically snapshots of intensity profiles picked up, scattered intensity profiles picked up by a series of detectors along the periphery of the light scattering envelope. So as the solution goes through this flow cell, we get these instantaneous snapshots of intensity against angle for that solution. And if this is then coupled to a concentration measurement device, like a refractometer or a spectrophotometer, if we know the concentration, we know the scattering intensity envelope, we can get a measure of the molecular size. There we are. This is the light going through the flow cell, and you've got all these detectors at various angles around the periphery. And from this, because you're measuring a snapshot with the bigger molecules coming off and then smaller molecules, from the columns, we can get a distribution of molecular weights with this technique. And this is an example of some mucin glycoproteins that Tony was talking about last week. These are colonic mucins. This is the native mucin molecular weight distribution. You see molecular weights very large, ranging from 1 million through to over 10 million. Then if we reduce the molecule, that is, we break all the disulfide bonds, which are holding chunks, we call subunits of the molecules together, using disulfide bond breaking agents. And those of you that have done STS Page before will know one of them. Remember a molecule called mercaptoethanol or dithiothreatol? We'll do this when we do our own electrophoresis lecture with Glad next week. These diol reducing agents are the most obnoxious, smelly molecules about. It's not the worst. The worst molecule we've encountered is one called TEMED, which has It's horrendous smell. But anyway, that's something else. So these thiol, that means these sulfide bond breaking agents. If you treat mucins with these, you get a reduction in molecular weights. Then if you use, you digest the molecule, which breaks the peptide bonds in exposed regions of the molecule, which are not covered by sugar, using, I think it's pepsin or trypsin, you get a further reduction in the molecular weight. This is an example of the use of SECMALs to look at the structure of mucins, and it has a high molecular weight limit of about 3, 4, 5 million, whereas DLS can go up to huge sizes, viruses, bacteria, very large types of molecule. Although when you get to very large particles, you've got to be careful about the type of dynamic light scattering theory which applies. Okay, this is what a modern instrumentation looks like. This is what we got over in the NCMH at the moment. And what you have here is, this is the light scattering photometer. So all this stuff is boxed up. Then you've got a concentration detector at the bottom, but also included here is, you can actually see here we've got the columns outside that temperature cabinet. You can see the separation columns here, and this is a viscometer, believe it or not. It's an online viscometer called a differential viscometer, which is linked up directly to the light scattering instrument and the concentration detector. So as the column separates the molecules in the solution, it goes through sequentially the light scattering detector. So you get the light scattering signal as a function of angle and concentration, from which you get the molecular weight of each molecular species going through. And then with the largest molecules coming through first. And we also measured the intrinsic viscosity with this device here. And then we measured the concentration. So obviously we got to correct for the time lag between the different instruments. Once that's done, we can get the intrinsic viscosity and as a function of the molecular weight. So you get molecular weight distribution and intrinsic viscosity distribution. That's a very powerful combination when dealing with understanding the properties of linear types of polymeric molecules, which we've just been talking about in the previous lecture. Okay, just a brief revision on the viscosity. So relative viscosity is just the solution viscosity divided by the solvent. Greek symbol eta, where the R stands for relative. And the reduced viscosity is just the relative viscosity minus 1 divided by the concentration. So the reduced viscosity has units of the reciprocal of concentration, which are mils per gram. So we've done that. And the intrinsic viscosity is the limit of that should say the reduced viscosity as concentration goes to 0. And I gave you the various, where are we? Yeah, forms of that equation before. This is that Markowing-Kuhn-Saccharada equation. Again, we mentioned this before. A is 0 for globular molecules, 0.5 to 0.8 for coils, and 1.8. That's the limit for rods. And we talked about how we can use this method, for example, for looking at the stability of various formulations by looking at either the change in intrinsic viscosity or change in reduced viscosity. with time. And this was looking at the stability of chitosan formulations at different storage temperatures. I think the interest of that was in its use as a nasal drug delivery agent. It's a very sensitive function of conformation. We've done that. So, for example, you want to look at a protein denaturing. This is the usual values you get for a globular protein, about 3.4, 3.5. As temperature increases, the molecule denatures. And why does it form a plateau here? That's why it's fully denatured. You can't get more denatured than fully denatured. That's why it does that. But this is this device here. This is this differential. It's called a differential pressure viscometer for measuring viscosities on line. So you don't measure flow times. You haven't got a time detector in here. What happens is you have it's sorted out in a way, so you're comparing the flow of solvent through a capillary at the same time as comparing the flow of your solution. And instead of measuring flow time differences, you measure what's called a pressure drop difference. And the relative viscosity is related to this pressure drop, delta P. IP is the inlet pressure. So from this formula here, you can get the relative viscosity in the same way you can get the relative viscosity from the flow times in the standard viscometer, YouTube Oswald viscometer setup we talked about last time. So it's a clever way of measuring viscosities automatically, coupled from the outflow from an SCC column. And if you go through your notes, we usually measure the viscosity at a finite concentration. And then we convert that to an intrinsic viscosity using we measure the relative viscosity at a finite concentration. And then you get the intrinsic viscosity from that formula known as the Solomon-Sayuta approximation. So you need to go back to your notes we did on viscosity in an earlier recording of this and see where that comes from. So by using the Solomon-Sayuta equation, we can get instantaneous, almost intrinsic viscosities coming out as the solution passes through the capillaries in the viscosity device, having come from the light scattering device. So if you couple these two together, allow for the difference in time lag, you get the intrinsic viscosity and molecular weight as a function of the elution volume. So it's a very clever combination of methodology. And this has been used, for example, to measure the intrinsic viscosity of antibody formulations, which may have the presence of aggregates. When we do the antibody biotechnology module, you'll see what the issues are with monoclonal antibodies and the presence of dimers, trimers, and other things, which can be a nuisance in terms of the quality of the antibody, but nuisance in terms of characterization of the antibody. So this is an IgG3 type of antibody. We haven't done these yet, but we'll do these in the antibody module. And there's our monomer with an intrinsic viscosity of 9.9 mil per gram. And here's some dimer, trimer, small amount here. We managed to separate away the monomer peak from the dimer, trimer. And that's important because the intrinsic viscosity is an important conformation parameter, along with parameters like the sedimentation coefficient from sedimentation velocity and a parameter called the radius of gyration, which we get for these types of molecules from X-ray scattering. This is another one here. This is a mutant IgG3, which is, see, this one's got a long hinge region. This is a mutant one, which has had its hinge region shortened. And you can see the reduction in the intrinsic viscosity, but you've got the extra aggregate here. So that's an example. So here we are. The blue signal here is the viscosity signal. The green is the light scattering signal at one angle we've chosen here, which is at 90 degrees. And the red is the concentration signal measured using a refractometer. And the retention volume has been corrected for the time lag between the different that one, and that one. That's the light scattering, that's the viscosity, that's the concentration. So it's all been lined up. Right, this is what you can do with this thing. By combining these different conformation parameters together and using a type of modeling called, remember bead modeling I mentioned before? Bead shell models. What's this thing here? Get rid of that. Yeah, it's better. Okay, this is published in Biophysical Journal some years ago. This is Yanling Liu, Emma Longman, who was a former MSc student with us, the old ABT course. It's linked up with, she was an MSc student with Pfizer, and we were looking at the effects of doing the mutations on IgG3. So this is the conformation of the IgG3 molecule with a long hinge, determined with bead shell modeling. And by combining viscosity, this is small angle X-ray scattering to measure the gyration radius. This is the viscometry measuring the intrinsic viscosity. And remember the Einstein-Simha shape function, which has a value of the spheres 2.5. Remember, Einstein made that mistake. So this is a similar parameter from X-ray scattering. You can get it for light scattering, but it's only works for very large molecules, whereas X-ray scattering is much more suited for looking at the RG gyration radius of macromolecules of this size, and then the sedimentation coefficient and the corresponding shape function here. So by combining all these measurements together, you can get an idea of the orientation of the domains. See if it was a blob or extended. This is the bead shell model for IgG3, and this is the wild type. This is the bead shell model for the hinge deleted IgG3 molecule. So that's an example of combining different measurements together, viscosity, with any sort of impurities removed using the SECMALS viscosity device, together with sedimentation and with X-ray scattering to get something which you couldn't get with X-ray crystallography. We can get that. Okay, what's happening now? Why is that locked? Right, the final thing, the analytical autocentrifuge before we go across to the NCMH. I'm not going to go into any great detail here. We'll go into more detail when we do the antibody biotechnology module, because it's a gold standard method for looking at the purity, aggregation state, and conformation of antibodies. So we do that in some more detail when we do the antibody biotechnology module. So this is what the inside looks like. I think I showed you this. No, this is the monochromator. So you put your rotor in the ultracentrifuge, and then you put on this monochromator here, which is used for the UV absorption optical system, which is tuned if it's proteins, usually to 280, because that's where the aromatics absorb, tryptophans, tyrosines, and things. Yeah, we did that last week with the spectrophotometers and things. Yeah. Okay, we've also got this, which is a laser attached to the monochromator. So this shines laser light through a different cell, and then this is for using what we call interference optics or refractometric optics, which can be applied to any type of macromolecule, not just proteins, where the monochromator is limited to usually proteins and nucleic acids. Based on, you know, the absorbance in the UV, and also the absorbance in visible, if it's a colored protein in solution, like hemoglobin, myoglobin, and vitamin C. Yep, good. Right, now there's two types of sedimentation experiment. This is the, I'm going to show you this. This is the beautiful cell, centrifuge cell, which we put the solutions in. Just a little, pass that around. See the ejection ports, you see the sector strips, see the two channels, one containing the solution and one containing the reference solvent. So this is the sector for the solution channel. After a period of time, under the influence of a centrifugal field, if you're looking down on this, what happens because of the centrifugal field? The molecules will sediment through the solution, yeah, and eventually get to the bottom. It doesn't all pile down straight away. Well, it will do if it's all big particles, but for a protein, even at 50,000 RPM, DNA polysaccharide, it takes a long time, it takes hours. It's a slow process, slow. High G-force, 200,000 G, slow process. And what you do is you scan across this at time intervals, the boundary, and that boundary will move slowly, okay, towards the bottom of the cell. So you can use absorption optics at 280 nanometers or interference optics, the refractometric optics, to get this boundary. And the rate of movement of the boundary per unit centrifugal field gives you the sedimentation coefficient, which is a measure of the size and shape of the molecule or assembly. Like a ribosome. So ribosome, 70S, 30S, 50S, whatever. That's how fast it sediments. Not sedimentation rate, but sedimentation rate per unit centrifugal field. Because how fast it sediments will also depend on how fast you're spinning the rotor. So that's why we use sedimentation coefficients, because it allows for the different speeds people use. Okay, sedimentation coefficient. And then you have the other experiment where you don't sediment quite so fast. Instead of 50,000 RPM for an antibody molecule or whatever, for a sedimentation velocity experiment, measuring the sedimentation coefficient, which is a function of the size. The bigger the particle, the faster it sediments. the more asymmetric the particle, the more friction it experiences, the slower it will sediment. Right? So if we want to use this for measuring molecular mass or molecular weight, then we have to have some sort of calibration. Alternatively, which allows for confirmation or correction of confirmation, we can run at a lower speed, say at 15,000 revolutions per minute. And what happens here is, as this stuff is piled down to the bottom, an opposing force called diffusion tries to push it all back, because this is thermodynamically unfavorable, having all the ordered so that all the protein is down there and all the liquid is here, all the solvent is here. So what happens is, to minimize the free energy, diffusion processes try to push everything back so it's equal concentration. You've got sedimentation trying to push everything down there, and diffusion trying to push everything back so it's not all down there, but all uniform concentration. It's two opposing effects: sedimentation and diffusion. So at very high speeds, sedimentation wins. The sedimentation forces are much greater than the diffusive forces. So all that happens is that this boundary becomes a bit broader with time. But at low speeds, the two forces become comparable. And what happens is that they come to equilibrium. So after a few hours, it stops moving. And what happens is you have a low concentration at the solvent meniscus, building up gradually to a high concentration at the base, and that remains stationary. When the forces have sorted themselves out, that's how it happens. It doesn't band. You don't get a band. You only get bands if you use what's called a density gradient sedimentation procedure, or use cesium chloride density gradients. But in a straight solution, you just get a low concentration building up to a high concentration at the bottom. This is sedimentation equilibrium. The forces happy, they come to equilibrium. It's minimum free energy, so everything is fine. It remains stationary through time. And because it's static, there's no movement. So there's no shape effects, no friction effects come into play. This is purely a function of molecular weight. The weight average molecular weight. All right, because it depends on the weight concentrations of molecules. So if you can measure that, you can get the molecular weight. If you measure that, you get the sedimentation coefficient. And if you know the molecular weight from that by combining sedimentation coefficient with molecular weight, you can work out what the conformation of the molecule is. And there's various software about for doing all this. The most popular is a program called SedFit. This gives distributions of sedimentation coefficient and molecular weight. If you know, remember this, the sedimentation coefficient molecular weight relationship, S equals K double prime M to the power B. Yeah, that's the power law equation. So you can get the molecular weight if you know the conformation type and correct for diffusive effects. But using sedimentation equilibrium, you can get the average molecular weight using M star, or now SedFit M star, because this has been built, the MSTAR algorithm here has been built into the SEDFIT program. So there's a SEDFIT MSTAR program now, which gets the molecular weights for you. And another one called multi-sig as well, which is useful. So we can get molecular weight. If you've got an associating system, monomer, dimers, trimers, and things, you can get the strength of the association via a parameter called the association constant. That's a quantitative measure of the strength of association or interaction between molecules. Right, these are what's called SEDFIT distribution plots. This is what we call a G of S plot, straight sedimentation distribution. This is IgG4 preparation after a freeze thaw. You can see the different, this is the monomeric species here, and you can see the different species of different molecular weight here. So this is not a very pure preparation. The manufacturer would want the molecule to be 99% higher than 99% monomer, and not with all, this is only 75% pure. The rest is aggregates. This is another, or you can do a C of S plot. C of S plot where you correct the distribution for diffusion effects. So you get a much sharper peak for the monomer. This is another antibody, IgG1-3. This is it's called a glycoform of IgG1. And you can see this antibody is a bit better. It's 80%. Can you all see that? It's 80% pure, but it's got some, again, some higher molecular weight components, probably dimers, and higher molecular weight components here, but also some smaller component. Maybe this is a Fab or FC fragment, which is broken off. So again, this is a very quick way of assessing the quality of your antibody molecule. We'll do this again later on. And you can convert the sedimentation coefficients into a molecular weight distribution based on the knowledge of the conformation or assumptions of the conformation. There's that power law equation which we've been talking about, which can convert this. So this is a relative molecular weight distribution based on that conversion formula. So you get a quick idea, relatively quick idea of the purity and molecular weight distribution or size distribution from the sedimentation coefficients, which can be interpreted in terms of molecular weight distribution for your solution. Yeah, that's just a bit of research we did a few years ago where we this is all based on basically spherical types of conformations, but we extended this to cover general types of conformation types of polymeric systems, including linear types of molecule, rod-shaped, coil shapes. And using this theory, you could convert sedimentation coefficient distributions for complex molecules like glycoconjugates into distributions of molecular weight. Again, using this is a rather complex form of that equation I gave you before, linking the sedimentation coefficient with molecular weight. This is for converting the distribution G of S into a distribution of molecular weight, but that's the power law coefficient here, where B is 0.4 to 0.5 for a coil, 0.15 to 0.20 for stiff rod, and about 0.2 for a spherical conformation. And this is the conversion for the two extremes of a random coil, 0.4 and 0.5. You can see how broad the molecular weight distribution of this material is, going up as far as 200 million molecular weight. So this, what's this? Right, and this has been integrated into the SEDFIT procedure, which we'll talk about when we do the group mini projects. This is a conversion for human gastric glycopolypeptide based on a random coil model, and that's the distribution of molecular weight. Again, very large distribution, millions of molecular weight. And then conformation zoning. Yeah, course 2 will finish in a second. This is, again, what we talked about in the last lecture, about the different zones: stiff rod, flexible rod, semi-flexible coil, random coil, globular or branch type of molecule. How do you find out what type of conformation zone or qualitative conformation zone that you have? Well, what you do is you combine the sedimentation coefficient with molecular weight information. This is actually the mass per unit length, and this is the concentration dependence of the sedimentation coefficient parameter. So by combining this data together, you see whereabouts on this conformation zone plot your molecule lies. So you measure the sedimentation coefficient parameter here. This is the mass per unit length and the concentration dependence of the sedimentation coefficient. It's got those three values. And then you find out where on this plot your molecule lies. Say if it lies in there, then you just say, oh, your molecule's a zone C molecule. Or if your parameters S, ML, and KS end up with your point lying inside zone B, then it's a zone B molecule. So by measuring these three parameters, you can get a qualitative idea of the conformation of your molecule with this conformation zoning. And then to get a proper quantitative picture of the flexibility, this is again for linear types of molecules, you measure the persistence length. And for the persistence length, you use a program called Hydrodynamic. And what Hydrodynamic does is it takes data sets of from the secmoles, it takes the data set of the intrinsic viscosity and molecular weight. So that gets fed into this program, Hydrodynamic. You then add your measured sedimentation coefficient, and if you have it from light scattering, your radius duration, that all goes into this what's called global fit program called Hydrodynamic. And then you get this beautiful rainbow type of plot. And from the minimum in this plot, which the Hydrodynamic program calculates, it'll work out from this contour plot. So you're looking for a dark blue color. That's the minimum. From the minimum, you get an estimate of the persistence length and the mass per unit length. So for this particular molecule, which is guar gum, it has a persistence length of about 9 nanometers, or 8 plus or minus 1. These are different polysaccharides here, locust bean gum. So 8 or 9, would you say that was a flexible molecule or quite a stiff molecule? What were the limits we had for the persistence length? Went from 2 to 200. So this is at the low end. So you think this would be quite a flexible type of molecule. Okay. So we'll do this in some more detail. But when we do the polysaccharide structure next week, we'll go into this in a bit more. All this has been as a quick overview in about an hour of what these different techniques do. We've done viscosity in some detail already. We've already done, also we'll do the ultracentrifuge in more detail when we do the antibody sure. And then when we do the group mini projects, you'll see how the DLS and SECMALs give information about molecules. You don't need to know the detailed physics behind all these methods. That's not what's required for biomolecular technologists, but just an idea how these techniques work and the sorts of information they can give you, how they complement each other. I mean, some of these methods give the same thing, like the weight average molecular weight. And that's why we call them orthogonal techniques. Fancy name the field uses, orthogonal. It doesn't mean right angles, it just means different ways of getting the same thing. And you see, in biomolecular science, the crystallography, crystal structure is this, that's it. Or diffraction, the DNA structure is this, and that's it. But when you're making measurements on solutions, it's what we call it's soft data, because sometimes you're wondering whether it depends on the instrument or who is operating the instrument, or do you get the same answers if you use light scattering to get molecular weight, the weight average? Do you get the same answer you get from sedimentation equilibrium? You should do. And if you do, that's good. So that's what pattern people like. That's what the FDA likes. It likes these orthogonal measurements to give the same answer, because you can have confidence then. But the fun starts when they don't agree. And that's what research is all about. Science is about them trying to explain that and then coming up with solutions. Okay. So that's why it's useful having different methods which give different information, but sometimes they overlap in terms of the information that they give, and that's what you'd like, because you want that reinforcement, that confidence in the data. Yeah, I mean, you don't treat data with suspicion always. You treat data with a little bit of caution, I think, but not skepticism. Not so that you sort of faces down to the floor all the time and no, can't be that, can't be that. You've got to use a bit of common sense, and sometimes the engineering approach that ought to offend engineers making approximations. It's got spherical this, rounding numbers off and all this to get an idea how things operate. The FDA and the regulatory authorities usually demand more than that. They demand reliability, accuracy, reproducibility, and if different techniques give the same answers, and if not, Why not? The more complex it gets, the more difficult it gets. Okay, any questions on that before we wander across? Okay, I'll just stop the record. I think that's gone.